In 2006, Harvard University BioVisions made a video entitled The Inner Life of the Cell. The video was one of the first videos that animated some of the amazing molecular machines that are now being found in cells. The overwhelming impression of design of the cell literally leaps out of the video at you. Since the intelligent design of the cell is readily apparent in video for all to see, Dr. William Dembski, one of the pioneers of the intelligent design movement, would, circa 2007, show the video in some of his talks on intelligent design. When Harvard BioVisions found out about Dr. Dembski using the video in his lectures, they warned him not to use the video. Their effort to stop Dr. Dembski was futile, since the video soon went viral on the web and anyone with access to a computer could download the video and watch it whenever they wanted to, and see for themselves the amazing design that is readily apparent in the cell. Anyway, fast forward to 2013. In 2013, apparently trying to undo some of the damage that was done by the Inner Life of the Cell video, Harvard BioVisions then made another video entitled Inner Life of the Cell Protein Packing. In that 2013 video, Harvard BioVisions tried to make the inner workings of the cell look as ugly, chaotic, and haphazard as possible. As you can see in the video clip, they tried to make the inner workings of the cell look as accidental and therefore Darwinian as they possibly could, so as to try to dispel any impression of design in the cell that they had inadvertently created in their first video. And here is a New York Times article on the protein packing video. In the article, Carl Zimmer states that in the 2006 version, we can't help seeing intention in the smooth movements of the molecules. But of the 2013 video, he said the molecules of the cell flail blindly in the crowd. Our cells work almost in spite of themselves. In the following article from 2014, Dr. Jonathan Wells takes issue with Carl Zimmer's description of molecules flailing blindly in the cell and states, but that's not what the biological evidence shows. In fact, kinesin moves quickly with precise movements to get from one place to another. Moreover, in the following article, Bruce Alberts, who was a two-time president of the National Academy of Sciences, stated that we have always underestimated cells, but at least we are no longer as naive as we were. Instead of a cell dominated by randomly colliding individual protein molecules, we now know that nearly every major process in a cell is carried out by assemblies of 10 or more protein molecules. Moreover, in the following 2016 paper, it was found that crowding in cells doesn't hamper protein binding as much as they thought it did. In fact, finding a lack of collisions in the crowded cell was a counterintuitive surprise for the researchers. Specifically, they stated, One would think collisions between a protein and other molecules on DNA would slow it down, but the system is so dynamic it doesn't appear to be an issue. And the following 2017 paper found highly complex architecture of the human cell, of which Dr. Hunter stated added more detail to the fact that the workings of the cell are far from random. In the following article studying photosynthesis, the authors state, the biological systems can direct a quantum process in astoundingly subtle and controlled ways, showing remarkable resistance to aggressive random background noise of biology and extreme environments. As well, Dr. Granville Sewell, who is a professor of mathematics at the University of Texas El Paso and who has studied the Schrödinger equation in detail, states that the Schrödinger equation is itself critical for life, and goes on to state that if elementary particles interacted by bouncing off each other like tiny balls, obeying classical Newtonian laws, chemistry would be dead. And indeed, the billiard ball model of atoms and molecules, which was apparently used as the foundational basis for Harvard's chaotic protein packing animation, is not even the correct model of atoms that is used anymore. The billiard ball model is basically a fallacious 19th century relic which is no longer regarded as the correct model for depicting atoms. Instead of a bumpy billiard ball model, we now have a far smoother quantum cloud model of atoms, as you can see on this timeline. As well, in this modern picture of atoms, you can also see that atoms are far smoother than was originally depicted in our early billiard ball models of atoms. In the following video, at the 24:30 mark, you can see a close-up picture of atoms that will clearly get this point across. Here's a short clip from the video.
Over 100 years after its discovery, we can actually see what an electron might look like by using the computer technology it created.
One of the neatest pictures that sums up the quantum world is the one where a ring of atoms has been made as a little fence. You can see the waves of what used to be thought of as quantum particles sort of filling this quantum corral, as it's called. They can't escape. They're stuck inside there, and the waviness is kind of frozen there and can be photographed. That's something that the quantum pioneers would have loved to see.
Schrödinger would have said, way cool, dude.
Don Eigler at IBM has used a quantum microscope to make pictures of atomic surfaces. Each step in this picture of a surface is just one atom high. On top of the atoms, the wavy pattern is caused by a sea of electrons.
These are just electrons which are trapped in the surface layer, but within the surface layer they're free to move around. These electrons are waves. And the waves, when they move, they sometimes bang into features on our surface, like the step edges or the individual atoms which might be sticking out of the surface. And when a wave bangs into something, it reflects off of that thing. And when you have a reflected wave adding together with the incoming part of the wave, it sets up what we call a standing wave. These are regions where there are large oscillations which are fixed where they are in space, and regions where the oscillations go to zero.
I've always felt that the wave function was just a description of a reality, and the reality was deeper. It was a particle, and the wave function was just something mathematical up in a physicist's head. To actually see a physical wave function rippling across the surface is rather disturbing. If I was really honest, I'd probably have to tell you that I need to go back and reassess the way I've pictured the world.
Well, I'm probably somewhere in error, or maybe I'm just a heretic. I don't believe in this wave-particle duality mumbo-jumbo. I think it's mostly just the leftover baggage of having started off understanding the world in terms of particles and then being forced because of the quantum revolution to think of the world in terms of waves, and we're stuck with this dualistic way of looking at these very small particles. Don't even think about them as particles. Electrons are waves, and if you think of them in terms of waves, you will always end up with the right answer. Always.
Moreover, in the following 2014 article, Jim Al-Khalili, hopefully I pronounced his name right, who is professor of theoretical physics at the University of Surrey, states that as 21st century biology probes the dynamics of ever smaller systems, even individual atoms and molecules inside living cells, the signs of quantum mechanical behavior in the building blocks of life are becoming increasingly apparent. And here's a short clip from a video where Al-Khalili drives that point home.
Millionth of a meter. My area is the atomic nucleus, which is the tiny dot inside an atom. It's even smaller in scale. But this is the domain of quantum mechanics, and physicists and chemists have had a long time to try and get used to it. Biologists, on the other hand, have got off lightly, in my view. They are very happy with their balls and sticks models of molecules. The balls are the atoms, the sticks are the bonds between the atoms, and when they can't build them physically in the lab, nowadays they have very powerful computers that will simulate a huge molecule. So this is a protein made up of 100,000 atoms. It doesn't really require much in the way of quantum mechanics to explain it. Quantum man begged to differ with this idea. Erwin Schrödinger, he of Schrödinger's cat fame, he's an Austrian physicist. He was one of the founders of quantum mechanics in the 1920s. In 1944, he wrote a book called What is Life? It was tremendously influential. It influenced Francis Crick and James Watson, the discoverers of the double helix structure of DNA. To paraphrase a description in the book, he says, at the molecular level, living organisms have a certain order, a structure to them that's very different from the random thermodynamic jostling of atoms and molecules in inanimate matter of the same complexity. In fact, living matter seems to behave in its order, in its structure, just like inanimate matter, cooled down to near absolute zero, where quantum effects play a very important role. There's something special about the structure, the order inside a living cell. So Schrödinger speculated that maybe quantum mechanics plays a role in life.
And in confirmation of Al-Khalili and Erwin Schrödinger's contention that life acts like inanimate matter cooled down to near absolute zero, in the following experiment, it was found that protein molecules do indeed act like inanimate matter cooled down to near absolute zero. And in the following paper, which found that the human eye can detect the presence of a single photon, the researchers stated that any man-made detector would need to be cooled and isolated from noise to behave the same way. And in the following article, the authors found that the long-standing mystery of protein folding can be easily explained if protein folding is regarded as a quantum affair. And the following paper also confirmed Erwin Schrödinger's contention that life is based on quantum mechanical principles. And as if the probability of finding a functional protein from random sequences of amino acids was not already an insurmountable difficulty for random Darwinian processes to explain, as these references illustrate. As if all that was not bad enough, quantum proteins make this insurmountable difficulty for Darwinian processes that much worse. In the following paper, it was found that the possibility of finding even one biomolecule that is in the quantum critical state by accident is mind-bogglingly small and, to all intents and purposes, impossible, of the order of 10 to the negative 50 of possible small biomolecules, and even less for proteins. And in the following articles and pictures of flagellum, you can see for yourself that the molecular machines of the cell are not nearly as bumpy and chaotic looking as Harvard's protein packing video had falsely portrayed them to be. As well, in the following paper, it was found that the flagellum motor does not belong to classical mechanics, but to quantum mechanics. In the following article, subtitled How Biomolecular Machines Can Generate Non-Trivial Quantum States, the authors state that entanglement can be maintained even in the presence of very intense noise. The authors then go on to state that this reverses the previous orthodoxy which held that quantum effects could not exist in biological systems because of the amount of noise in these systems. Environmental noise here drives a persistent and cyclic generation of new entanglement. As well, besides proteins and molecular machines, it is now found that DNA itself does not belong to the world of classical mechanics, but instead belongs to the world of quantum mechanics. In the following video, at the 22-second mark, Dr. Riper shows why high temperatures do not prevent DNA from having entanglement, and then at the 24-minute mark, Dr. Riper goes on to remark that practically the whole DNA molecule can be viewed as quantum information with classical information embedded within it. Moreover, in Penrose and Hameroff's infamous OR model for quantum consciousness, they claim that microtubules would display quantum mechanisms. Confirmation for quantum mechanisms in microtubules was found here. Moreover, the evidence that quantum mechanisms are at play on the macro level of the human brain, besides just the micro level, is revealed by the following. In the following article, it is noted that multi-electrode recordings have revealed zero time lag synchronization among remote cerebral cortical areas. As well, evidence suggesting that quantum mechanisms are at play on the macro level of the human body itself is also revealed in the following article, where it is revealed that a subject perceives a sensory stimulus on the skin at the moment the skin is touched, before the stimulus reaches the brain and before full deliberative consciousness occurs. As well, it is important to note that both quantum entanglement and quantum coherence are non-local, beyond space and time effects. As well, in the following paper, it was found that the greater number of particles in a quantum hypergraph state, such as what we see with quantum biology, the more strongly it violates local realism, with the strength increasing exponentially with the number of particles. And although many people like to imagine that particles with what are termed hidden variables are somehow the cause of what Einstein termed to be spooky action at a distance, a new experiment has now found that entangled objects, whether subluminal, superluminal, or even instantaneous, do not cause each other to behave the way that they do. Thus, since quantum correlations are now shown to be completely independent of any time and space constraints and cannot possibly be explained by reference to the particles themselves, then one is now forced to appeal to a cause that is itself not limited to time and space in order to explain these quantum correlations. For the Christian theist, this should not be surprising. The Bible has always held that God holds all things together. As well, it is interesting to note that quantum entanglement can be used as a quantum information channel, and that this quantum information is conserved. That is to say, it cannot be created or destroyed. Besides providing direct empirical falsification of neo-Darwinian claims that say information is emergent from a material basis, the implication of finding non-local, beyond space and time, and conserved, cannot be created or destroyed, quantum information in molecular biology on such a massive scale in every DNA and protein molecule is fairly and pleasantly obvious. That pleasant implication, of course, being the fact that we now have strong physical evidence suggesting that we do indeed have an eternal soul that is capable of living beyond the death of our material bodies. In the following video, entitled Quantum Entangled Consciousness, Stuart Hameroff states that it's possible that this quantum information can exist outside the body, perhaps indefinitely as a soul. Well, that's the end of the video. Thank you very much for watching. And again, all papers and videos referenced in this video may be accessed in the link that is provided in the video description. Thanks again.